Welcome to our journey through human physiology. Today, we are going to explore the gastrointestinal tract, which is arguably one of the most fascinating and vital life-sustaining systems in your entire body. We will uncover exactly how it breaks down the food you eat, how it perfectly balances massive amounts of fluid every single day, and how it uses ingenious microscopic structures to maximize nutrient absorption and keep you alive. To begin our exploration, let us imagine your digestive system as a highly advanced factory. But instead of putting things together, this factory is an automated disassembly line. It takes complex, bulky raw materials, like a cheeseburger or an apple, and systematically breaks them down into microscopic building blocks. These tiny building blocks are then shipped out to power the rest of the city, which in this case is your entire body. The gastrointestinal tract is essentially a long, continuous, winding tube that starts at your mouth and ends at your anus, acting as the ultimate recycling and processing plant. Now that we understand the overall purpose of this disassembly line, let us look at the specific machinery involved. Formally, the gastrointestinal tract is known as the alimentary canal. It consists of primary organs, including the mouth, pharynx, esophagus, stomach, small intestine, and large intestine. Alongside this main tube, there are accessory digestive organs like the salivary glands, liver, gallbladder, and pancreas. These accessory organs secrete essential enzymes and fluids into the tract. The entire system works through a beautifully coordinated series of mechanical movements, like chewing and stomach churning, combined with chemical processes using powerful acids and enzymes. With all these organs working together, you might wonder why this specific system is so critical to your immediate survival. Every single cell in your body requires a constant, uninterrupted supply of energy and structural materials to survive, repair, and function properly. The GI tract is the sole gateway for these life-sustaining nutrients and water to enter your internal environment. In a real-world clinical setting, if a patient's GI tract fails, they can quickly suffer from fatal malnutrition and severe dehydration. Furthermore, it acts as a massive physical and chemical barrier against harmful bacteria and toxins that we inevitably swallow. Beyond just extracting nutrients, this gateway plays a massive role in keeping you hydrated, which brings us to fluid balance. To truly understand how hard the GI tract works, we must look at the massive volume of fluid it processes daily. It is not just about the water you drink. Your body actually secretes gallons of its own digestive juices to help break down food. The total volume of fluid absorbed by the intestines must equal the total fluid entering the tract, minus the small amount lost in waste. Understanding this balance is crucial for treating patients with severe dehydration. To see exactly how much work the intestines are doing, we can express this physiological balance mathematically. The formula states that V-abs equals V-ing plus V-sec minus V-x. Let us break down what each symbol means. V-abs represents the total volume of fluid absorbed into the bloodstream, measured in liters per day. V-ing stands for the volume of fluid ingested through drinking and eating. V-sec represents the volume of endogenous fluids secreted by the body's own glands, like saliva and gastric juice. Finally, Vx is the volume of fluid excreted in the feces. If secretions increase dramatically or absorption fails, excretion skyrockets, leading to life-threatening dehydration. Before we move on, Let us see this formula in action with a concrete example of a healthy university student. We are given that the student ingests 2 liters of drinking water and half a liter of water hidden in food, giving a total ingested volume of 2.5 liters. Next, we sum all the internal glandular secretions, which include saliva, gastric juice, bile, and pancreatic fluids. Adding these reveals a massive 7 liters secreted by the body itself. We plug these totals into our balance equation, subtracting the tiny 0.1 liters lost in feces. The final answer shows the intestines absorb an incredible 9.4 liters of fluid per day to prevent dehydration. Now that we have seen the massive volumes of fluid handled by the gut, let us look at how it physically manages to absorb all of it. The small intestine is the primary site for nutrient and water absorption. However, it is confined within the limited space of your abdomen. In biology, whenever an organ needs to do a massive amount of work in a very small space, it folds its tissue to create more surface area. To maximize nutrient absorption, the inner wall of the intestine is not smooth. Instead, it features multiple levels of structural modifications that drastically amplify its functional area. To understand this biological folding, let us examine the specific structures that line the intestinal wall. If the small intestine were just a simple, smooth tube, it would have a base surface area of roughly 0.33 square meters. But to survive, we need much more than that. The inner wall has large circular folds that increase the area significantly. On top of those folds are tiny finger-like projections called villi. And on the cells of the villi are even smaller, microscopic hairs called microvilli. Each of these structures sits on top of the previous one, multiplying the total surface area available for absorption. Because these structures build upon one another, we can calculate their cumulative effect using a simple multiplication formula. The equation states that A total equals A base times F1 times F2 times F3. Conceptually, this models the physical reality of the intestinal lining. A total is the final functional surface area. A base is the initial surface area of the smooth tube. The variables F1, F2, and F3 represent the respective multiplication factors for each structural modification: the folds, the villi, and the microvilli. If any of these structures are damaged, for example by a disease, the corresponding factor decreases, drastically reducing the total area and leading to malabsorption. Let us apply this amplification formula to find the true functional area of a human small intestine. We are given the base area of the simple tube as 0.33 square meters. The circular folds multiply this initial area by a factor of 3. The finger-like villi multiply it by 10, and the microscopic microvilli multiply it by 20. We first multiply the 3 amplification factors together, giving us a massive total amplification factor of 600. Finally, multiplying the base area by 600 yields our final answer. The effective surface area is 198 square meters, which physically ensures every nutrient molecule has a high probability of being absorbed. To truly appreciate this incredible system, let us look at a complete visual overview of everything we have discussed. On the left, you can trace the continuous muscular tube from the mouth, down the esophagus, into the stomach, and through the winding small and large intestines. Notice the accessory organs, like the liver and pancreas, connected to the main tract. On the right side of the diagram, we have a magnified view of the intestinal wall. You can clearly see the large circular folds, the finger-like villi projecting outward, and the tiny microvilli forming the brush border. This diagram beautifully illustrates how macroscopic anatomy and microscopic structure work together to sustain your life. As we wrap up our overview, let us review the most important concepts we have covered today. First, the gastrointestinal tract is a highly specialized, continuous processing tube designed to extract nutrients through mechanical and chemical digestion. Second, we learned that it secretes and absorbs massive volumes of fluid, and maintaining this balance is critical for your hydration. Finally, we saw how the intestine uses folds, villi, and microvilli to amplify its surface area by 600 times, ensuring absolutely nothing is wasted. You now have a strong foundation for understanding how each specific organ performs its unique digestive magic.